Hello and welcome to our newest conversation of Let's Talk About Hanford. Tonight's topic is about pollinators. To cover this, we have invited our own George Peck for a second presentation on this very vast topic. My name is Ana Alvarez, and I am in the communication section here at the Nuclear Waste Program. We started the Let's Talk series back in 2020 to cover Hanford related topics at a high level. We will continue doing so as long as we have interest and participation from you guys. On that note, thank you to all of you for being here tonight. Regarding tonight's presentation, we ask you to please put any questions that you may have or any comments into the chat box on Zoom or on Facebook at any time. After the presentation by our guest speaker, George, we will have a live question and answer with all of you. You can ask your questions directly by unmuting yourself, or if you prefer to just ask the question in the chat, please add those at any time. If you'd like to ask your question out loud, you can put your name in the chat box or raise your hand. We really encourage questions and comments into the chats at any time, but please make sure you're gearing those questions to tonight's topic, which is Hanford pollinators. If you have any questions or comments that aren't geared to tonight's topic, please email hanford@eci.wa.gov, and we will be very happy to follow up with you. I think that with all that being said, I will welcome our speaker tonight, George Peck, and take it away, George. Thank you for being with us today.
Thank you, Anna. It's great to be here. I think we're ready for the first slide as soon as that can start. I can show the slides myself or oh, good. Dana's going to be helping me. Perfect. Great. So tonight I want to talk to you a little bit about the pollinators of Hanford and South Central Washington. Next slide. So tonight we'll talk a little bit about what a pollinator is, just a quick overview. Then we'll transition to important crops and pollinators in South Central Washington. The third topic is crop pollinators up close. The fourth topic is Hanford pollinators up close. And the fifth topic would be a bee box study and also something about monarch butterflies. And then we'll close the session tonight with advances in pollinator detection technology. And I'll also be sharing some important pollinator resources with you. And this slideshow will be posted on the internet so that you'll be freely available to look at these sources and links at your leisure. Next slide. Okay, what is a pollinator? Well, a pollinator is an animal that moves pollen from the male anther of a flower to the female stigma of a flower. And so here on the right-hand part of the slide, we have the plant anatomy. This is a cross-section of a typical plant. The anthers are the dark orange structures. There are 4 of them in the drawing. Those are the male structures of the plant. And then the central structure is called a stigma. And so with pollination to be successful, the pollen grains have to move from the orange, dark orange anther, also called the stamen, to the pistil. Now that can happen by just wind, but usually it happens by some sort of an insect or perhaps other invertebrates like hummingbirds, like bats. And the pollination process helps bring about the fertilization of the ovules or the female gametes. And so the ovules are down at the bottom of the stamen. They look like, maybe a bunch of little peas in a pod. And so the pollen grains actually have a cell that will literally eat down through the tissue of the stamen and find the gamete. And so there's more on this that I can share with you, and there's links throughout the talk that will elaborate on that. Insects that pollinate include bees, bumblebees, wasps. They include butterflies and moths, flies, beetles, and others. And I mentioned bats and hummingbirds. And then also humans can artificially pollinate, of course, in agricultural settings and in a laboratory to experiment with different outcomes. Next slide. This is a diagram I shared with you back in November when I talked about insect diversity in general. What I want to point out here is that there are species of Hymenoptera, and the Hymenoptera are an order of insects that includes bees and wasps. But there's also the fly order called Diptera, the Lepidoptera order, butterflies and moths, the beetle order called Coleoptera, and those are the main pollinators that we'll be focusing on tonight within this talk. Next slide. Now let's talk a little bit about the pollinators of Hanford and South Central Washington. But before I do that, I just want to notice that I'm hearing a number of interactions with the chat. So if there's any questions, I'm going to take a break at slide number 12, and maybe we could save our questions till then. But if there's a burning question, don't hesitate to bring it up right away. So there's 20 crops that constitute most of Washington's agricultural production, which is about $2.5 billion a year. At least it was in 2023. Now, many factors affect pollination of agricultural crops, especially bee pollination. The host plant has to be attractive to the bee. The weather has to be not too hot and not too cold. And then the habitat has to be suitable. It can't be over water. It can't be very, very windy. There has to be certain areas where the bees can develop nests. Now, honeybees, we humans create our own nests for honeybees, but every other bee has to find a nesting area. So that's why we have to have suitable habitat. For fruits or seeds to develop, pollination must occur at blossom time. Blossom is when the plant produces flowers, the flowers open, and pollen and nectar are available for the pollinator. There are nearly 1,000 species of bees in the Pacific Northwest, which is quite a lot of species to be tracking, and we'll be learning a little bit about that tonight. But only a small number of bee species are useful in the pollination of orchard crops, which we'll be talking about tonight. Now, pesticide use and the loss of appropriate nesting habitats have reduced wild bees considerably. And most of the pollination of commercial orchards in Washington is dependent on what we call the European honeybee or the Western honeybee. The scientific name is Aphis mellifera. It means bee that brings the honey. Next slide, please. Here are the 20 main players in Washington, Central Washington agriculture. So we have apples, and when you see an asterisk next to the crop, that means it's required, it requires a pollinator to be able to set fruit and to be productive. Potatoes, second most important crop in South Central Washington, doesn't require a pollinator. Alfalfa does not require a pollinator when it's grown for hay, for animal feed. However, there's a large portion of Washington that is producing alfalfa seeds for production of alfalfa all over the world. And to produce an alfalfa seed, you do need a pollinator. Wheat is windblown pollen. Hops, it's windblown. Grapes, it's pretty much windblown. Cherries absolutely need a pollinator. Pears need a pollinator. Corn's windblown. Blueberries, raspberries need pollinators. Canola, it'll help the yield of the canola if we have a pollinator, but it's not required. Chickpeas, peas, barley, mint, all of those are wind pollinated or self-pollinated through a little bit of vibration because of the wind. Pumpkins, yes, they do need a pollinator. Lentils, no. And finally, peaches and apricots need pollinators. So that rounds out the 20 crops. And if you look at the Google image, you can see Hanford directly in the middle of the image. And you see that it's surrounded by large areas of irrigated agriculture. So there's a lot going on out there and lots being produced. Next slide. Okay, I created this table to give you a sense of the important pollinated crops that we'll be talking about tonight and how many acres there are that were harvested in 2023, how many pounds were produced, and the value on the market of that crop. So let's take a look at apples, for example, 173,000 acres. The production, well, over 7 billion pounds of apples produced in 2023 just in Washington state. And the value of those apples was nearly $2 billion, shipped all over the world. Delicious, famous Washington apples. Second place with cherries. And third place was pears. Fourth place, blueberries. Fifth place, raspberries. Sixth place, pumpkins. Seventh place, peaches. And then finally, one of my favorites, apricots. And part of my career has taken me to the Prosser, Washington area. So I included a picture, an aerial photo of WSU Irrigated Agriculture Research and Extension Center. And this is just outside Prosser. And so that's an important place where research on pollinators and agricultural crops throughout southern Washington goes on as we speak. Next slide. Okay, this is where we're going to start to name pollinators of each of those important crops. For apples, we see that honeybees are the most common or mostly pollinated by apples, but there are other things that can and do pollinate apples. Orchard bees, bumblebees, solitary bees, wasps, flies, and beetles. All of those can pollinate apples. But in Washington, out in our fields, it's primarily the honeybee that gets the job done. And those honey from, good to know. Apis mellifera in general, worldwide, has 8 species. And all of them originated millions of years ago in the Afro-Eurasia area. The one that we domesticated and that we used in Europe and in South America and in North America, evolutionarily, it was roughly in Eastern Africa that we can trace its ancestors to. So it was domesticated that we know of 2,600 years ago during the Egyptian Age, during the Bronze Age. Moving forward, it was brought to North America and the East Coast in 1622 by German colonists. And then finally brought by ship to the Pacific coast of North America in the 1850s, probably to help with crop production during the gold rush. It's the dominant pollinator in Washington cropping systems. It's a eusocial bee. It's very social. It has a very hierarchical structure. There's jobs for everyone in the hive. Primarily, it's the females that get the job done. There are male bees, but their sole job is just to mate with the queen. Worker bees cooperate to find food, and they use a pattern of dancing within the hive called a waggle dance to actually communicate regarding the direction and the distance to patches of resources. Like they can dance a certain way, and all our sisters say, "Oh, you mean the patch of daisies that is 200 yards east of us? Come on, let's go check it out." So truly, it's an amazing system that bees have. And they're extensively used for fruits and veggies when needed. And their production is worth billions of dollars. They add 9% of the value to every crop in the world. Next slide. So moving to less known and less popular pollinators, but important that we have these pollinators, especially with climate change and with many of the diseases that are attacking our Western honeybee. And that is the blue orchard bee, Osmia lignaria. Now, this is a cosmopolitan worldwide solitary bee. Um, there's a number of facts here. I'm just going to read through them for you. These bees emerge when it's about 58 degrees Fahrenheit. So they overwinter and sleep until things get warm. When a female and a male emerge from a hole, they mate immediately. The female matures over a few days as she feeds. The ovaries become ready for oviposition. And then that female will seek a hole to make a nest. And so as you can see in these diagrams, that central lower diagram, a blue orchard be provisioning a nest. So a nest can be anything from, say, a bamboo shoot to a soda straw, depending on the size of the bee. And when they provision the nest, I would draw your attention to the lower left-hand side of the slide, where they make cells. They partition cells, and they provision each cell that they make with a little bit of pollen mixed with nectar. And when they get enough of that material in a single cell, they'll lay an egg upon that material, and then they'll close up the cell with mud and saliva. So they're really good at working with mud, working with saliva, and fashioning kind of little apartments for their babies. And when those babies mature, they will push themselves out the end of the tube and emerge as adults. And they go through what we call holometabolous metamorphosis, where there's an egg, then there's a number of larval stages, then they finally pupate into a pupa, and then that pupa rearranges itself and emerges as a full adult bee. It's the same way with butterflies through the chrysalis, and it's the most advanced system in insects. So there's the blue orchard bee for you. Many people are trying to work with this bee in a number of orchards as an alternative to honeybees. Next slide. Now here's another pollinator that is worthy of mention, and it's used throughout the world in special cases and being introduced more and more places. There's many, many species of bombus throughout the globe, 29 in Washington. They're fairly social. They form small colonies with a single queen and 50 to 100 sisters. There's division of labor, just like in the honeybee. But after the season ends, the females are mated, and then they usually overwinter underground. The nest looks much like what we see in the lower right-hand corner of the slide. It's fairly loosely aggregated. Those white structures are the waxy cells containing the larva of the bumblebee. There's also some what they call pots of honey. So they store honey, and they also carry pollen. And so again, in most bees, the diet of the larva is a mixture of honey and pollen. They forage fairly close to the nest, and they use something called buzz pollination. And this is really important for tomato plants, but also for peppers and blueberries. It's a special technique where the bumblebee kind of disengages its flight muscles away from its wings, and vibrates its entire body. And that vibration is strong enough to shake the pollen down from the anthers to hit the stamen and create a pollination success story for tomatoes, for peppers, and for blueberries, something that a honeybee cannot do. And then they regurgitate their nectar and the pollen to feed to the developing larvae. And that's their life cycle. Next slide. Here's an interesting story. It's important for Washington. I bring it up here, and it shows you very clearly what researchers are doing to help with alfalfa seed production. So in this right-hand photo, we see two researchers checking the health of an alfalfa leaf-cutting bee condominium. So thousands of tubes are placed within an alfalfa seed field, and the alfalfa seed bees are reared in their thousands, released, and when the females mate and then begin to provision for their young, and then when they seek a nest, they will enter one of these holes, and they, again, just like the other bumblebees, will partition off single cells, add the material for the eggs to develop upon, and create a successful pollination to produce alfalfa seed. And it's really amazing that if you have thousands and thousands of females coming into a condominium area, how can they possibly identify their exact nest? They do. So insects have incredible olfactory smelling capability, and also memory of where, with certain cues like the direction of the sun or shade, but they're able to find their nests. So there's truly, in that very, 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 very small bee, an amazing amount of mental acuity. Next slide. So I'm going to pause at this point and ask if there are any questions, because we're transitioning away from agriculture into the Hanford entomology. Okay, no burning questions, then let's just go right into the next report that I'm going to be covering. So I talked a little bit about these reports that I'll be talking about more in depth today during my talk back in 2023 in November. This particular biodiversity study was 5 years long, 1994 to 1999. Almost 4,000 specimens were collected. There were over 1,500 species level identifications made. 49 species of butterflies, 318 species of moths. And of all of those species, there were 41 species and 2 subspecies that were brand new to the scientific community. No taxonomist had ever found these particular species, so that's always an exciting thing. And one of the key take-home points of this study was that Hanford writ large has an assemblage of microhabitats that are large enough to support pre-European insect and plant fauna. And that's why the Hanford Reach is so important. It's been preserved for years. And if it continues to be preserved, it'll be a refuge among all that agriculture we saw, where we can get a glimpse of what things looked like before the 1850s, when large-scale agriculture came to South Central Washington. Next slide. Here's a map showing the sampling area on the right-hand side. The black triangles show the particular areas that were sampled for insects over time. Sweep nets were used. They actually have tarps that are set underneath plants, and you can beat the insects out of the plant and into a tarp. It doesn't harm them, but it's a very good way of figuring out what's out there. Hand picking, light traps, and pitfall traps were used. Insects were mounted and labeled just like good entomologists do, and this work was done at Pullman and at WSU Tri-Cities. There were certain insects that were focused on in 94 and 95. It was leafhoppers, true bugs, beetles, bees and wasps, flies, moths, and butterflies. And then in 96 and 97, that changed a little bit. There was nighttime trapping, so more moths were trapped. True bugs were trapped, also more beetles. Lacewings are very easy to trap at night, and others. And those trappings were used, they used a mercury vapor lamp placed on a bed sheet to bring insects in for those trapping events. And then finally in 1998, pitfall traps were used at sites, and those are very good for collecting ground nest or beetles that are mostly terrestrial ground crawling beetles. All right, next slide. So the results of this particular biodiversity study that I want to focus on here in this slide are the pollinators. There are 16 orders of invertebrates within the study identified, over 1,500 individual insects identified to species level. All 5 main pollinator groups were present. The Xerces Society that you see there in bullet number 3, they're an invertebrate conservation society that are based in Portland, Oregon. And they take scientists and knowledge from throughout the world, and they've got advocates to help people understand the best way to preserve insects in all their shapes and forms. So the 5 important orders that are going to be discussed tonight are the beetles, the flies, the hymenoptera, which are bees, ants, and wasps, and then butterflies and moths, and then the numbers that were found in the given study. There were 43 species that were new, and there were also 142 new occurrence records. So that's important because we realize now that Hanford has a lot of insects that maybe were present in other places in Washington, but until this study, we didn't know that they were present in Hanford. Lots of good things were learned from that study. Next slide. Okay, we're transitioning now to a Hanford pollinator study. This study was done it's about now almost 8 years ago. And there were 4 study sites that were investigated. And each of those study sites had a different type of a habitat. I mentioned this back in November. It was grassland, it was rabbit bush dominated, there was a mixed shrubland, and then there's mature sagebrush. So a variety of habitats brings a variety of pollinators. Next slide. There were over almost 2,000 total bees collected during this survey in 2017. There were 5 bee families observed. And here's where it gets interesting. The sweat bees were the most dominant species of bees out in Hanford. Over 1,000 of those were collected during this study. The next most important family that was collected was the Megachilidae, which are mason and leaf-cutting bees. There were also Apidae. The family Apidae was collected, and those include bumblebees and honeybees. And then there are also mining bees, or scientifically family Andrenidae. And then there was finally some plaster bees or polyester bees, really interesting primitive little bees, and they're named the family Coletidae. Next slide. So here is your whirlwind tour of Halictidae, the sweat bees. These are cute little bees. You don't really see them if you're not looking for them. They're small. Some of them are beautifully emerald green colored. And there's a particular genus called Lasioglossum that was the dominant genus within Hanford in this study. And these are between 2 and 8 millimeters long, so very small compared to a European honeybee or a bumblebee. But in Washington, there's over 100 species. So now we know there's quite a few species within Hanford as well. There's another genus called Agapostemon, and we see in this study that the 281 of those were captured. There's approximately 10 of those species that are known to be in Washington, and all of those species have this emerald green coloration. They look like little, they're beautiful. Yeah, got to see them to believe them. And then finally, the genus Halictus, this study recovered 25 of those. There's approximately 10 species of that genus in Washington, and those are a dusky black with striping and easily identified by their wing venation. So there's your whirlwind tour of Halictidae. So we need to move on and learn about these other families that were discovered at Hanford. Next slide. The mason and leaf-cutting bees, the megachilids, again, nesting in cavities, single cell provisioning, much like that orchard bee I showed you earlier, where cells are stacked one after another and the brood pushes through when they're fully developed. Pollen and nectar feeders, as most bees are. 273 were collected in this study, and there were the genus Megachilidae, 19 of those. There's over 1,000 species of that one globally. And that includes the alfalfa leafcutter bee. Even though that was brought to the United States, there were already native megachilids within the United States. Then there's a tribe called Anthid Anthidiini, and there's many species of those globally. 65 of those were discovered. Um, and there's another tribe called Os Menini. 13 of those were recovered in this study, and over 1,000 species globally. And then there's one recovered in this study called Lithargopsis. And as you look at these montages of all of these different bees, you might be saying, well, I'm already like overwhelmed. They almost all look the same. And yes, as an entomology student, that's exactly how you feel if you're ever asked to identify what bee am I looking at. But when one does begin to identify an insect, there are distinguishing characteristics that one looks for. And so immediately a beetle is a beetle is a beetle, a fly is a fly is a fly, a bee is a bee is a bee. But it turns out that there's what we call dichotomous keys that you can go through and actually bring your identification down to a species level if the species are well known and identified. And most insects, not all, there's many that we don't know about, but many are well known. So there's your taxonomy lecture for tonight. Next slide. Cuckoo bees, carpenter bees, digger bees, bumblebees, and honeybees are all in the family Apidae. And so they're all social to some degree, except what we call the orchid bees. In this study, 156 were collected. There's a family called Eucerini. They're longhorned bees. And I bet if you look carefully at that photo montage, you can figure out which one is the longhorned bee. There's also the genus Diastea, and these are oligolectic. That means they are very specialized on a certain plant with certain pollen. And that's important because if you have Hanford and you have a bee that's dependent on a certain plant at Hanford, if the plant goes extinct, the bee goes with it. So it's important for us to, as ecologists, to know these relationships so we can make statements about conservation. Anthophorini, those are digger bees, love to dig in the soil. They're cute, white, fuzzy little bees, and 18 of those were recovered in this particular study. Bombus, the bumblebee genus, only 8 were recovered. Not quite sure why, but there's many reasons. Hanford's a pretty austere environment, and so to be able to rear a large bumblebee colony, it may be better to head to the west side where there's more rainfall and habitat more conducive to being a bumblebee. And finally, Apis, the genus of honeybees, only 3 of those were recovered. We don't know if they were mellifera, but that in of itself tells me that all of the thousands and thousands and thousands of bees that are within, say, 100 miles of Hanford pollinating apples and pears and apricots and peaches, that just a trickle of those ever actually get blown into Hanford. So it's its own unique island of special organisms. Next slide. Mining bees, the andrenids. So these are cute, tiny bees, 2 to 10 millimeters. And there's a genus called Perdita, and they're also extremely specialized on pollen. So you take their pollen source away, the bee perishes. Little tiny fairy bees were very common, over 87 of those collected. And then there's also the genus Andrena. They're mining bees. They're also solitary and not well studied at all. And that's the general story about bees, is that we want to have successful agricultural systems, have successful crops. So we spend a lot of money getting the honeybee successful, but very little money is spent on these smaller bees that are part of our ecosystem, but mostly ignored. All righty, I'm at slide number 22, and I'm just going to ask again if there's any burning questions. Let's go ahead and try to answer 1 or 2 of those.
George, I do have a question.
Uh-huh.
I wanted to know, in that study that you said that they found a different or a new type of bee, was that particular and specific to the Hanford site? And how does this happen that there are new species? Is it that there's a mixture that occurs between two different ones? I'm just curious about that, how that works.
The first part is yes, they found new species on Hanford. And that just means that it doesn't mean that they're unique to Hanford. It just means they found them on Hanford. And maybe if someone had collected outside of Hanford, they'd realize that maybe they're up in Othello or something, or even up by Grand Coulee. It just happens that someone collected really well on Hanford. Hanford has had financing, so maybe it's just a matter of we got a lot of money to sample, but there's so much more to learn. The second half of your question, creation of new species. So with bees, these are species that evolved over millions of years. So it's not like since agriculture's been in Hanford that a new bee has had a chance to evolve. It's really, really, really constrained over millions of years. So when I said that there were, say, 100 new occurrences in Washington, that means that if that was a bee that's been found, say, up by, oh, I don't know, the Mental Valley, way up north by Canada. They know it's up
Mhm.
there now. Now they know it's in Hanford too, and now and someone found it in Pullman, someone found it in Walla Walla, and now they know it's in Hanford. Um, so I think the big story really with these small, tiny bees is no one has time to go look. And they're out there, and we just we're just hoping that, as people become more and more interested, as they become more and more concerned about the world with climate change, that more and more people will realize that, I got some free time. I'm going to do some naturalizing. Maybe I'll have an app on my phone and I can just snap a photo and I can upload it into an artificial intelligence system. And Professor Gonzalez, Professor Smith, and Professor Wing at the university will be very, very happy to collect. I mean, citizen science can be all part of this. So I'm just rambling. Did that kind of answer the question a little bit?
Absolutely, absolutely. And
Okay.
can I have a follow-up question? Is there like a database where there are like all the species listed, and whenever there's a new one, they just add it to this database? I mean, I don't know. Is there something like that in existence right now?
Good question. A worldwide database. I can't think of anything off the top of my head. Um, that being said, Hymenopterus or bee specialists, they have their own careers and their own worldwide meetings. Maybe every 4 years they meet wherever
Hmm.
they would meet, share information. They have their own journals. There's Journal of Hymenoptera, say, and it may be a monthly journal with full of articles that would connect people in a way that they share information. So there's that type of scientific sharing. But to answer your question, Anna, I don't know of any worldwide database that we could Google right now. That would be a question I'd have to research and get back.
Awesome. Thank you, George. Let's
Okay.
continue. Appreciate it.
Yeah. Okay. Let's now go to slide 23. Okay. This is the last slide we have in the up and close. What we found in Hanford, the Cletidae, only 3 of these were found. They're very unique bees. They line their nests with a cellophane saliva. They're solitary bees, but they like to aggregate, and they're very primitive bees. So they don't have a scopa or a pollen container on their back legs like the honeybee does. So they have to carry all the pollen they collect in their crop, which is basically their stomach. So not only do they regurgitate the honey to their larva, but they regurgitate the pollen too. And there's many species worldwide, but only 3 were collected in our study of Hanford. But those are an important part of the ecosystem, and we know they have a place and a purpose. Next slide. Okay, now we're going to talk a bit about a nest study that was done at Hanford. This was done in 2019, 2020, and 2021. Some habitat restoration was done in the 200 area of Hanford, and it was done because a water line was dug and vegetation was destroyed, and vegetation was regrown over the water line. And because the federal government and the laws and regulations of our entire nation require us to mitigate for habitat loss and to understand species present and impacts to species, the Hanford folks put out bee houses. And so let's talk a little bit about that. They had occupancy rates on all 3 years within the bee nest boxes of 30%. And in the photographs, you can see the line there, the placement between 200 West and 200 East, where the individual traps were located, or not traps, but bee boxes. And then in the lower right hand, you see a blue tub. That's last year's bee box that has the bee babies in it that are emerging. And so we see that the hole in the top of that tub next to those rocks is a place where last year's bees can emerge and participate in the next year's breeding cycle. And then on the other side of that, we see, of course, the present bee house on that T-post. Next slide. The results from these 3-year study, occupancy rates, only 10 in 2019. That's because they started in July. It was a late start, but in 2020 and 2021, they had pretty good occupancy rates. They speculate that the bee tribe Os menini was the main user of the nest. That's in the family Megachilidae. But weather, resource abundance, and unique bee species ecology. Some bees are liking certain plants or certain weather patterns, or the winds were very, very strong and it was just too much for the bees to be productive. Many factors over many years can depend and influence the bees that are present. And the authors of this study ended their study by saying that this could be a key habitat restoration tool as Hanford continues to be cleaned up over years. And I just direct your attention to these upper photos here in the upper right. You can see that there are a place where the leafcutter bee has completely emerged. There's a hole up there with a little caption next to it. And then there's also nests where the brood, the larva, the pupa are still incubating. And so emergence will be coming soon. Next slide. Okay, I'm looking at my clock and I realize I've gone quite fast, but there's still a lot to cover. And we've got things we could be talking about next time we meet. We haven't talked about the butterflies, wasps, flies, and beetles as pollinators. What about pesticides? What are the factors that affect the population dynamics, and how can we conserve them? So let's take a quick look at this photograph here, and I've circled a wasp. So we know that this is a milkweed and many species are utilizing it. So let's see how quickly we can finish the talk off. Let's move to the next slide. So this is the importance of pollinators and monarch butterflies. And I see that I've used a lot of your time already. So I think the best thing that I should do is just leave this story of the monarchs for another time when we can talk about them in great detail and really serve them with good justice about what they do and how important Hanford and other places in Central Washington are to the conservation of these world travelers that started in central Mexico and move all the way up to Canada and make that journey every year. And it's truly amazing. Many people are astounded by it, myself. So let's leave that for another time. So can we skip maybe 3 slides ahead to 29. I promised you something about the future of pollinator monitoring. If I could summarize this in 10 seconds, there are camera systems and there are computer algorithms that are able to analyze a photograph of a given insect and make a best guess with artificial intelligence, AI. So there are definitely investigators. This is the University of Pennsylvania that are looking for collaborators that will bring this to the next level, where instead of having to go out in the field every day with traps and all of the paraphernalia that you need to do entomology, there can actually be just cameras taking photographs and a way to, in real time, through many monitoring systems throughout a habitat, be able to get real data for conservation and other purposes. So if there is more interest in the audience, of course, this will be available to you with links to those sites. Next slide. This is, you see a panda bear there. This is just a slide to basically tell you two things. First of all, there are downloadable free apps, for example, Seek by iNaturalist, that you can get on your iPhone right away or your Droid right away. And they are helpful in identifying photographs of species. But I will caution you that an AI system, all AI systems, whether it's for self-driving cars or insect identification, can make mistakes very rapidly. And this particular study shows where scientists published results of an adversarial photograph. They tested an AI system to see if it was reliable. So they took a photo of a panda and then they added in a tiny, tiny bit of interference. And so they asked the AI system, what is this interference? And the AI system said, well, it looks like a lot of dots, but my programming says that I'm going to say it's a nematode with 8.2% confidence. Okay. And then when they added the panda at a 57.7% confidence through the AI system with the nematode interference, then they asked the AI system again to give them a true identification. And the AI system said it was a gibbon, which is a monkey. Now, you and I know with our beautiful acuity and our insight and, identification abilities as a human, it's a panda. The point is AI systems, and we hear about them all the time. We just need to be really cautious with them and have a human checking the AI system for accuracy and precision. Okay, I think we're almost done. Next slide. So this is just free, fun websites to explore, both researchers in Washington and general interest sites about pollination, about pollinators, why they're important to the world, how everything really will collapse in terms of our agricultural food system if we lose all our pollinators. And then let's go on to the very last slide. These are essential resources on honeybees and other pollinators, including information on the Xerces Society and the USDA Agricultural Research Service, Washington State and Oregon State, all for you to explore. I think we're almost done. Do pollinators fit into the Hanford cleanup? Yes, is the answer. They do. Our laws mandate it, and we want to follow the law and make energy production possible, also simultaneous with habitat preservation. And so if you are more interested in that, you can read up on that on the Hanford site's ecological monitoring. So that is all I have tonight. I appreciate you allowing me to go way over time. And I'm happy to entertain questions. You can email me with more questions. And my job at Nuclear Waste Program is facilities transition. I'm working in Purex. I'm working in the 400 area. I'm working in the 300 area. And I'm working with the Department of Energy and Central Plateau Cleanup Company, with tribes, with EPA, working as a team to create conditions that will continue to push forward. But I'm always thinking of things outdoors and fun pollinators. So don't be afraid to bug me. That's
Awesome. Thank you so much, George. We appreciate having you for a second time.
oh,
And I heard we're going to have you here for a third time. So we're ecstatic about that one, about monarchs. I am going to invite now my co host, Ryan, and wonderful supervisor, by the way. He's going to join me in asking questions either from the public, or I think we had a couple questions for you, and then we can just wrap up. Ryan?
Yeah, so I think just like Andre Gracel said, if anybody has any questions on either Facebook or Zoom, just drop them into the chat, and we'll just get you started with some of the questions we had. George, I had a question listening to your presentation. You kind of covered this, but you talked about there being about 1,000 species of bees here in Washington. And sorry if you did cover this and I missed it, but how many specific species of bee have been found at Hanford again?
in Hanford, this study that we investigated, that we looked at today. So there was the study with in the biodiversity study between 1904 and 1999. You're asking for an exact number. I don't have an exact number of bee species. There were papers published from the study that may give you a number. So I'd have to look at those studies to know for sure. So I don't have the exact number of bee species there. In general, 1,000. Given that it's Central Washington, it's an arid habitat, which always has more species. It's kind of strange. And then the idea that it's an undisturbed habitat, that it's basically as it was from the beginning of time. And then thinking about the pollinator study itself, where I think they didn't always identify it to species level. I think there were one or two cases where they knew exactly which species they had. So Ryan, just to be sure I have your question right, when I say a species, or when you ask for species, for example, Apis mellifera, the honeybee, that's an exact species that would be a count of one. Then we had a bumblebee, Osmia ligulera, that would be a count of two. Um, I'll just throw out a number because I don't have that information. There's probably close to 150 species of bees out there at least. And so I talked with Richard Zach by email about a week ago. Richard's a scientist at WSU that actually led the studies in 94, 95, 96, 97. And when he heard that I would be giving a talk on bees, he was excited and he said, "Oh, I'd like to get back out there and sample the bees." Because not only is it, well, it's fairly easy to catch them, but you may be asking around the world to find a person that specializes in bee identification. So, and there's a number, for example, Utah, Logan and Provo, Utah, have a huge group of bee experts. But it's one thing to grab them, and then there's another thing to find someone, volunteer to, yeah, I'll figure out exactly what those are. So have I given you a good enough answer?
Oh,
Hopefully.
that goes above and beyond. Yeah, so I wasn't expecting like a specific count. I was just more curious about compared to the state, maybe a rough number for the area. So that was super helpful. So, with that, I'll kick it over to Anna Gracel if she had a question, or if Dana did, or if we've gotten any in the chat yet. Anna Gracel, back to you.
That was just a follow up. That study that you say that ended in 1999, has that been the last one done in this regard? And are there plans to do new ones, more updated ones?
Um, Not that I know of. And I'm not sure what prompted Dr. Zach and the others to do that. The actual document, there are reasons to conduct those studies. And like I said, there's laws and so forth. And so in the grand scheme of things, if we were to propose to the powers that be, we want to do that study again, they I have a feeling they'd say, well, you did it 20 years ago. Not much has changed. Go ahead and do it, but we're not going to give you any more money. So we know Hanford's incredibly diverse. This gives us just a sense of how incredibly diverse these systems are because they've been left alone. And so you know, with that 94 to 99, that was the big one. And then the pollinator study, it was only looking at bees. So it wasn't looking at the full diversity of insects. So there's definitely a need to keep monitoring, and we'd have to show a compelling reason to the Department of Energy that it's, again, time to go out and take another good look.
Right. Thank you so much, George. I know,
Yeah.
Dana, there are any questions that you want to ask?
I did. I had a question when you were going through, George. You were talking about all the different types of insects that are pollinators. One of the ones you mentioned was a true bug. And how is a true bug different from a regular bug?
Okay. True bugs are what entomologists classify as heteroptera. Heteroptera means that their wing structures are heterogeneous. Optera is wing in the Latin, genius, heterogeneous. So those are considered true bugs. What is a great example of that would be a leafhopper. I don't have a photo of one, but they're small sap-sucking insects, and you approach them and they jump really fast.
Okay, I know those bugs.
Yeah. At this time of the year, there may be some out there in the parking lot of Ecology, box elder bugs. They've got mostly black body, but they've got a red triangle on their back. Those are also part of that true bug family. What's another good example? Brown marmorated stink bugs that have invaded Washington over the last 15, 20 years. Those are also of that family, Heteroptera. If you've ever had a whitefly infestation in your garden, those are Heteroptera, true bugs. Surely you know what an aphid is.
Yeah.
That is part of that large order called Heteroptera.
Well, then it begs the question, is there a family called fake bugs?
Oh,
If it's a
fake.
true bug, is there
Oh,
a untrue bug or a fake bug?
well, the true bug comes from entomologists hearing the general public saying, they're just bugs. They're just bugs. And that that really, that entomologists have, they just don't want everyone to just call it true bugs. So there's no such thing as a false bug unless you say like a centipede's a bug. And that's why the common names are useful to in some instances, but scientifically, globally, we have Latin binomials. So there's no confusion. If I say, I just saw a red stinky bug, and they'd say, oh, well, in where I come from in Germany, that's called a beer sucker bug or something. And someone else would say, well, down in Africa, they call that a tsetse bug or something. So there can be hundreds of common names. So for an entomologist, they I guess I'm answering a question. A true bug is only one order, and that's that order Heteroptera, and there's thousands of species of true bugs, but it doesn't mean that everything else that is in the class Insecta is a false bug. It's just that
They don't fall into the big family.
yeah, it's at that point when you're just like, that the professor is just going to say, do you see why using common names creates confusion? Because wherever you come from the world, it could be a different name for Aphis mellifera or Culex mosquito or something. I mean, so the precision of science requires us to drill down and use something in a Latin name rather than to rely on common names like a stink bug or a buzzy fly or I mean there's also a pill bug or there's
Roly poly.
a roly poly, yeah. But there's truly, there's a Latin binomial to describe that. And there's actually a way to classify it. It's not an insect, but it is an arthropod, and it sits inside a phylum called Myriapoda, many-legged. So there's a method to the madness, and when you start to study something as vast as invertebrates, you're thankful that there is method to the madness because it helps you to classify things and to find some sense about, okay, I just saw that bug. It's got piercing mouth parts. Its wings are folded in this such a way. It's got to be within this order. And then you say, well, and then it has this type of leg structure or this type of eye structure. And they say, well, that's within the family. So that's how an entomologist grabs an insect just right out of the garden and says, okay, family Heteroptera, and then can identify it just by site identification. And then eventually, of course, a book or an expert would have to take it to the species level because there's literally millions of the species.
Yeah.
Birds, that is fascinating. I have another question I wanted to ask, but I wanted to first again see. I think we have a quiet lunch tonight on the streams, but I wanted to make sure we had an opportunity if anybody had any questions to ask them away. The Zoom chat. I think we've got a few attendees on Zoom. You guys are more than free to either unmute or put anything in the chat if you have anything. It's okay if you don't have any questions. I just want to make sure you have the opportunity to. But so I guess while we're waiting to see if anybody does have any questions before we wrap up, George, one question I had was listening to your presentation. You talked about, and something that was kind of new knowledge to me was you were talking about the and I have a note here on my that I jotted down. You talk about the five types of orders of pollinators, and you said that all five of those orders were kind of discovered at Hanford, and you listed out. I think there was between a couple hundred to 300 types of those pollinators of each category, except one category had 50 or something like that. So I was just curious, does it matter if there's an unequal balance of pollinators? Does that have any kind of impacts? So for example, if you had a bunch of pollinators of four orders and then none of one, does that have any kind of impact over on the local habitat stuff, or is that just organizing them? I'm not sure that question makes sense.
Good question, Ryan. It depends on the habitat and the plants in the habitat. In a rainforest where there's lots of orchids, absolutely important to have unique pollinators that fit inside that orchid. They've co-evolved. They've got to have their own pollinator. If they die, the other dies. Then there's the other side of the spectrum where you've got something like a dandelion, which is a plant that's been transported all over the world. And anything can pollinate. You just walk across the top of it and then the pollen sticks to your legs. Then you fly to the next one and you just stumble across it and the job's done. So there's generalist situations or generalist strategies, and then there's those orchids that are just suited to only one pollinator. And there's kind of everything in between. The tomato family, it's called Solanaceae, and bumblebees are really, really specialized to create conditions to allow that pollen to shake down from the anther to the stamen. So there's other examples as well. There's a lot of plants that have a really deep corolla, and only bees and moths that tongue can extend over 2 centimeters can reach down in and get that nectar gift. And so only certain species are able to even attack that corolla to get what they want. And so they're co-evolved with only certain pollinators. So it's really a story of co-evolution, plant-insect interactions over millions of years that have created highly specialized relationships, and these relationships are most common in the tropical regions of the world. Does that help?
Yeah, that's super helpful. I love it.
Okay.
Okay, well, still not a lot on the Zoom chat, but that's totally okay. Means that I had a very good comprehensive presentation. But Anna Gracel, Dana, do you guys have any other questions before we wrap up?
Yes, I have one more, and this is probably going to be a long answer. So, I mean, I don't know how much of these you can also answer, but we briefly touched on, the risks and what is global warming, doing in these habitats. And so, if you can briefly tell us, especially on the Hanford side and how we can mitigate that, I think that would be good for participants to know as well.
Right. Oh, let's focus on Hanford. Let's focus on climate change. And what we know that as the climate warms, it's going to be more extreme. Rainfall will come faster and quicker. Winds will be stronger. Hot will follow very cold, will follow very hot. All of these things are not good for tiny bodies like insects. So it's going to be more stressful every single way for insects to survive in climate change. Their host plants that they depend on may become stressed as well because of all of the things I just talked about because of climate change. So that being said, there may be a tiny, tiny, tiny few insects, terrible insects like mosquitoes, that may actually have huge outbreaks because of heavier rainfall and more water in certain areas. And that creates West Nile health problems. So the world is going to be more challenging with the emissions that are going into the atmosphere right now in many, many, many ways. And so for Hanford, it's going to take committed people and committed agencies that really want to commit time and energy into studying how these climatic changes are affecting the plants and animals. And it takes data gathering. And I mentioned one of my slides, there are technologies coming available where there'll be probes, just set some camera systems out in the field for months on end and just collect that data. So there are going to be opportunities to make it easier to monitor greater areas and greater numbers of species. And so challenges and interesting developments. So and what I just said about Hanford, it truly means every insect all over the world. It's going to be more challenging to grow crops, to have crops adjust to more harsh, rapidly changing conditions. Every ounce of human energy and human intellect will be needed to find cooperative solutions for all societies as we continue to transition into these new areas of climactic issues. Yeah.
Thank you so much, George.
Yeah,
I
I
am
think
going to do a last call for questions, either Ryan or Dana or our chats, and if not, I think I'm just gonna wrap up, Brian.
I was gonna say I could ask you thousands of questions of bugs and we'd be here all night. So I'll probably hold any questions I have, George.
No
Okay,
worries.
so
I'm here. Okay.
thank you, George,
Thank
again
you.
for being with us tonight. And we would like to thank everyone for attending tonight's event and for just, being engaged with this topic that is so interesting. We will be bringing probably the monarch topic because I think George has a lot to say about that. And I think to make it justice, we should do its own thing. This is just a reminder for everyone that the event was recorded and it will soon be up on both our ecology Facebook page as well as our YouTube channel. We will also add all the slides that you saw tonight to our website. And I will encourage attendees to reach out to us if you have any questions at all and to stay tuned to future events on these topics about Hanford. George, if you want to share with us any final thoughts, I welcome those. And thank you again.
You've covered it well. Thank you.
Thank you so much. And have a good night, everyone.